Thank you for the introduction. Thanks for inviting me to this excellent meeting in a wonderful scenery. So I would like to show you today work that I started right in the beginning of the latest pandemic. And this was our big motivator. So we really thought of how we can use what we do on a day-to-day basis, at least in my lab, which is fluorescence-based single molecule spectroscopy, to the benefit of the pandemic, if at all. And if you can also compete with existing technologies and bring something to the table that helps the world, although we were kind of late with our response. But yeah, that's a side effect. So our lab does a lot of things, and as the title says here, life is short but interesting. So I would not tell you all of what we have on our buffet, but I'm going to focus mostly on what we developed, which is a flow virometer, which basically counts events, detection events of virus particles. It's not the first flow virometer in the world, but I think in means of specificity and sensitivity, we are competent in that manner. So I like to present my group and collaborators in the beginning, because I never know whether I'm going to reach the end. So I'm going to focus mostly on the work done by my student Paz and by my research associate Yair. And we have a very nice collaboration going on together with Torben Cordes, who's in the audience, and with the Israeli Institute for Biological Research. It's one of the few biosafety lab 3 institutes in Israel. So since we're working with live viruses, we have to work with them as well. So let me show you a little letter. It's written in Hebrew. Who in here knows Hebrew? Okay, so I'll translate what's written here. This was sent to us 2 weeks before this date. And largely speaking, what it tells you is that you either need to go home and start to become theorists for who knows how long, or work on emergency research related to COVID. And this was exactly 2 weeks after my lab was established and everything was installed. So I said, let's look at my drawer ideas that I never established and think whether we can come up with a quick and dirty experiment that will convince the rector in 2 weeks' time that we do have something on our plate that can be considered as an emergency project. That was the main motivation at that time. But we never knew that it would grow into something that is actually relevant and important. So I hope many of you know those lines and remember them. Those were the lines of waiting for PCR tests, but there were also lines waiting for the results of the tests, which took hours to days. And you know, back in the days, I thought to myself, okay, PCR, specificity and accuracy cannot be beaten. That's known. But on the other hand, people behaving like people in society, waiting for so long, and then maybe are positive and transferring their viruses to others. This is not so efficient. What could shorten those lines? So obviously, a few months later came those antigen tests, which are really rapid. We all know that. Okay, they are really reliable as long as you have positive results. If you have negative results, then you don't really know. And this reminds me of my old school computer science coding skills of an infinite loop, where somebody needs to be tested again and again and again forever until it is found positive. So that's, you know, that's nonsense. So I was thinking to myself, what can we do more that will be as accurate as PCR, but will be fast and reliable? So one idea that we had is maybe let's not try to quantify the genetic material in viruses, But instead, or in addition to, let's try to just count the virus particles one at a time and be very good at this. They're specific, selective, and also deal with the extremely low quantities of them in bodily fluids. So it's also about sensitivity. So viruses, it's a nano meeting. So viruses are nanoparticles, but very soft ones. I will touch base also on that in a few moments. They come at various sizes and various morphologies. They are quite complex. And unlike other biologically related nanoparticles, like vesicles, like other membranous particles, they are not as soft as them. So they are quite dense, mostly because they are packed with genes at extremely small volumes. So our hopes were that they will be as dense as inorganic particles, so that when we flood the surrounding of the viruses with a lot of fluorophores, fluorophores will not intrude into those viruses, and then we can see some signals following the contrast between what's outside and what's inside the viruses. So, but size is not enough. Size is not specific enough, because as you can see, there are many types of viruses of the same size, right? So obviously antibodies will add another specificity layer. So, and another thing that is important to remember is the sensitivity. So this graph here shows you the amounts in your endotracheal tract and also in saliva, which is the liquid that we wanted to focus on, 0 days after symptoms show up. So I hope you already know that symptoms show up about a day or two after you're actually infected. And the decrease in the amount is from about 1 million particles per milliliter in your saliva, all the way to about 1,000 after about 2 weeks' time, which back at the day in the beginning of the pandemic was the time you needed to wait until you were considered free of virus, right? And anything below 1,000 particles per ml were considered as non-symptomatic, right? But still possible to, possibly contagious. So just, you know, for the sake of magnitude comparison, we're talking about quantities that are from 1.6 femtomolar to 1.6 attomolar. Okay, so it's really low. What we were used to working in the lab are usually picomolars, right, using confocal spectroscopy. So we really need to think hard in order to not get into lots of false negatives, right? So the detection scheme that we need to think about should have high specificity, all right? So signals should report very specifically on what we see. We need also to count enough detection events that we're sure of, so that we get a very low p-value, means high certainty of what we found, and we need to do it at a fast time. But also important, you know, we're in a nano and quantum mechanical conference here, but we want to be as simple as possible to reduce also the cost of what we're coming up with. And we want to be also efficient. And so these things should be taken into account, because there are a lot of possible solutions, but many of them are not necessarily things that we can employ immediately, or not necessarily things that would be cost effective for whoever who wants to use them. So this is not the type of things that in normal times we thought of, but now it was. So many years ago, actually in 2009, and after it in 2010, there was this pair of papers from Widengren that came up with an approach, which is called inverse-fluorescence cross-correlation spectroscopy. So fluorescence cross-correlation spectroscopy, I hope some of you know it. I will just touch base on it for a moment. So you have noisy measurements. The noise comes from the low counts of your fluorescent molecules in a given effective excitation volume. The noise is not white noise, and therefore the autocorrelation of the signal includes some information about the amount of your fluorophores in average and about the mobility of those, or the mobility times of whatever you measured. But what we're measuring here is not labeled, fluorescently labeled molecules. What we're measuring, or hope to measure, is virus particles that are not labeled. All right. So instead, maybe let's not work in the dark where there are no fluorophores in the solution. Instead, let's fill the solution with a lot of fluorophores, totally irrelevant to the virus particle. And then let's add the virus particles. And hopefully, once a virus particle enters in the confocal spot, then it excludes some of the fluorophores that are excited in the confocal volume. And there will be a temporary decrease in the signal. All right. And obviously, the temporary decrease, its size, is proportional to the ratio of the volume of the particle to the volume of the excitation. All right. And then you can do also inverse fluorescence cross-correlation spectroscopy if you have two detection channels, one for the free fluorophore in solution, the other at a different spectral range for an antibody that is labeled, let's say, with a red dye. So now you don't only measure something that is proportional to the size of the particle, you also measure something that if it goes through the beam and it includes the specific antibody, that means that's a nanoparticle that is labeled specifically with an antibody to that virus and not necessarily other nano-sized particles that exist in our saliva. So, but this was done already in 2010, and I remember that because it was a really cool application, but we never had a reason to go to these scales, which are really nanoscales, right? We work at much lower scales, where the volume of whatever is emitting fluorescence is extremely low. You don't see anything about the noise of the constant signal of the fluorescence. But now we are in game because we have viruses, and they are about 100 nanometer in diameter. So the volume is not negligible relative to the excitation volume. However, this application was done on beads in diffusion, and the information is not taken from each and every dip or burst. It's taken from the whole data. That's what you do in fluorescence cross-correlation spectroscopy, right? So you basically need to take a lot of measurements, just a long measurement, and take the autocorrelation or the cross-correlation functions and gain some inferences from it. But there is also another technique, which is largely called fluorescence fluctuation spectroscopy, in which you really analyze each and every burst because it contains information. The width is really about how long it took for the particle to cross through the confocal spot, right? And it's related to the size and also to the viscosity, to the size of the particle and also to the viscosity. And since these are diffusing in a given excitation volume, you would expect that for a given particle of a given size, you will have a very wide range of values for the time it takes for the particle to cross through the excited volume. So maybe what we need to do is to work in flow. So largely speaking, the analysis is performed on all detected events, not on each and every one. There are many different signal duration and sizes per given size, and we're dealing here in a situation in which we want to take saliva that includes viruses and many other things. Each and every one has its own distribution of sizes, right? And so particles are moving in random paths throughout the laser focus. What we really want to do is to standardize the mobilities using flow. So, and we want to do it as much as possible in laminar flow, so that if we put our flow, our laser at a given position in our flow channel, it's largely speaking constant flow in given direction. So the only parameter that will influence how long will it take each and every particle size to cross through the effective excitation volume would be the drag of a given size of a volume. Right, the larger it is, the longer it will take it to cross through the effective excitation volume. So we're talking about low Reynolds number for large particles. So the concept that we came up with is the following. Let's take this idea that I've shown you now and take a one-channel microfluidics and put our effective excitation volume at the center of the channel. So these are channels that are commercial, they are extremely cheap, right? So it's following, what we thought about in means of, say, affordability. And just having a syringe pump, pumping continuously flow into the microfluidics. Here is our effective excitation volume. It's focused inside the channel. And imagine that you have those viruses anywhere. Okay, some of them cross through our beamline, our focus. Here is one that is unlabeled. So if it will pass through the effective excitation volume, it will give me a dip, just because it excludes some of the fluorophores in my confocal spot. And so it creates a dip, but it's not unlabeled. So it will not give me also a burst. So these are ones that include dips, but not bursts. But I have other ones that are labeled with the antibody. So while they cross through, they emit light at different wavelengths, which I detect. So I'm seeing the exclusion of dyes due to the inverse effect, because it excludes some of the fluorophores in my volume, but also emissions in the other channel of the antibody. All right, so this is what we thought. And this is just a simple scheme of what we came up with. It's a microfluidics with two-channel single excitation confocal scheme, right? And in a more specific manner, what we really aspire to is so that we will be able to differentiate between a correlative dip and burst. So this would mean that we found specifically a nanoparticle of a given size that we can quantify from the size of the dip. And also it has the specific antibody labeling it. But we want to differentiate it from the many other nanoparticles that might exist in our saliva that will give us just a dip. And the many antibodies that are unlabeled then are not labeling, because we pour just antibodies and we have free antibodies also in solution. So we would expect also some bursts without a dip. Okay, so that was the idea. So, and now the only thing that we need to do is to count those events, all right? So before we do that, we wanted to check whether we were able to standardize the size of the dips of the reduction in the signal relative to the size of the particles. So we worked with different beads, not yet biological particles, okay, of different sizes. And we tested the statistics of those dips. So largely speaking, what we saw is that there is some scalability of the sizes, the distribution of sizes that we get based on the duration of the dip, okay, but it stops at around 300 nanometer. We know why. This is mostly because the volumes become so low as relative to the magnitude of the noise of the constant signal from the fluorescence, right? But we can rely more on the duration of the bursts. These also give us scalability that goes all the way to 100 nanometer, right? So we can know something by looking at the width of the burst, at a given flow rate, for what's the size of the particle, right? What about separability? So there are many experiments we can do. One is in which we take a mixture of beads. One includes a specific labeling, the other is not. So 100 nanometer beads that are dye-labeled with red, and 500 nanometer beads that are just not having any dye. So here is one instance out of a long trajectory where we see 200 nanometer beads with dip and burst, and one 500 nanometer beads that is not. So we have separability based on the correlated event or non-correlated event, and also on the sizes of the dips that we get, right? But then came the real confusion. So we took viruses that we created on a tissue, all right? What you're seeing here are two types of viruses. On the left, what you see is the SARS-CoV-2 coronavirus after it has been neutralized to reach the BSL-1 level. This is done with performaldehyde, 2%, right? This basically has allowed our collaborators to send this virus to our lab from the get-go, right? And on the right, what you see is a horse virus. It's called vesicular stomatitis virus. It's not cross-contagion, okay? And it's not circular. It's more like a bullet shape, but in means of volume, it's just 8% above the known volume of the SARS-CoV-2. This was engineered to remove its G receptor protein and to include the spike protein from SARS-CoV-2. So in principle, it's still targetable by our antibodies, okay? To our surprise, what we found is that instead of seeing signals that are correlated dip and burst, we see signals that are correlated burst and burst. We checked in many controls. This is not crosstalk between the detectors or anything of that sort. This recurs again and again. So we had to rethink what we're doing, right? So here is the signal for the coronavirus. Here are the signals for the VSV. So what we're thinking that is happening is that unlike what we wanted, where really the virus particle is an opaque, highly dense nanoparticle, it's not. It contains some membranes that it included, all right? Because these viruses eventually are grown by budding out of cells, right? Instead, what we have is, from some reason, accumulation of our free dyes around the surface of the virus, so that while you have a constant fluorescence signal from your effective excitation volume, once a virus enters, it comes with a slightly higher concentration of fluorophores around it. So the effective amount of fluorescence while it crosses is higher than the constant level. So we are using it, and what we tested is 3 types of measurements. We knew from the beginning that our neutralization protocol with the performaldehyde is also making the virus cluster, all right? So what we did is we measured SARS-CoV-2 and VSV spike with the fixation protocol, and also the VSV spike without the fixation protocol, okay? So just to let you know, Israel also wanted its own vaccine. So the idea was to engineer this VSV spike and to use it as a vaccination, since it contains the spike protein, but it's not contagion at all. This succeeded until phase 2, but failed miserably at phase 3. But when phase 2 was done, we already had the RNA-based vaccines. So from the safety point of view, basically using it in my lab is like adding another vaccine, if you wish. But the most important part of this is that this one, unlike the VSV spike that is fixed, is not clustering. So it should give me the smallest signal, because it should not cluster. It should have its own volume, right? And So we measured a lot of those signals from those 3 measurements, and we put them on a scale relative to the beads that we measured. And they, largely speaking, they are showing the monotonic trend. But also, I hope that you can see that the ones that are fixed are including also a wider distribution, as also the beads include. So this also keeps an open question on why do we have different sizes of clusters, right? Yeah. So largely speaking, we got the specificity. Now we want enough detection events, right? So here's the idea. We can ask how many specific detection events we need at the shortest time possible. So our thresholds were, we want to measure for no longer than 10 minutes, and we want to get as much as a minimal amount of 100 detection events. So an alternative would be the question of what's going to be the minimal viral load that we can measure to reach these thresholds. So with laminar flow in our commercial 1-channel EBD microfluidics, what we got is we reached the level of 10 to the power of 8 particles per ml, which is not enough. We need to reach the level of 10 to the power of 6 to the 10 to the power of 3 particles per ml. And the reason is because really the cross-section of our effective excitation volume is much smaller than the cross-section of our channel. We're missing a lot of viruses. Actually, 99.999% are missing. We want to reduce the size of the analyte flow. So the idea was maybe to try hydrodynamic focusing. So instead of having 1 inlet, we will have 3 inlets and then focus our beam at the center of the chip. This was a design that was introduced in the past with a 90-degree junction, which is optimal, but it costs a lot to create it at large scale. So, you know, it brings you excellent reduction in the cross-section of the analyte flow. But we want to keep it simple and affordable. So instead, we're using a Y shape, which really means that the hydrodynamic focusing effect will not be maximal, all right? But it's very cheap. So here is an example of what happens when we open the flow from the side. Basically, flow buffer or whatever you want from the side at a ratio of flow rates that is higher towards the flow from the side. And we really reduce the cross-section to 30 micron. So here is just a scanning image of what happens to the particles where they flow. And this last bit was when we close the flow. So it's just adapting back. So what happens is obviously our particles squeezing into this small flow through, but on the other hand, they are accelerating. And sometimes they might accelerate beyond the time we wish them to go through our focus, because there is also the dead time of the detectors that we need to think of, right? So eventually we were able to show that we can do quite a lot of squeezing, and this is proportional to reaching the limit of detection of 100 events in 10 minutes for viral loads of 10 to the power of 4 particles per ml. Right, so with this trick, putting our effective excitation volume at the junction in a hydrodynamic focusing approach, we're able to be sensitive enough to reach the region that is biologically and biomedically relevant. And last but not least, simple and affordable, we use microfocal setup, which is costs a few hundreds of thousands of dollars. This is not so, doesn't make so much sense. We want to reduce the price and make it robust, also make it easy to use. So we work with the group of Torben Cordes, who is there at the back. And they have been able to produce not one, but several layouts of a 3D printed scaffold, which really economize the components and the whole solution. And it stays stable, and you don't need an optical table for it. This is how it looks like, at least the one that we use. It's really using laser pointer type, something like that, at the side, with two simple photomultipliers. And the cost of the whole prototype is about $8,000. So it's quite a lot of a reduction. And so I don't have much more time. So with that, I would just like to tell you that just compared to ELISA, which is another very sensitive but very classical and traditional technique of looking at not the virus, but the receptors on the virus. So an easy comparison would show you that at best, the sensitivity is down to the 1 femtomole or 0.1 femtomole in ELISA, while in our case, it can go down to the 1 zeptomole if you're using the hydrodynamic focusing approach. And the time it takes at the most sensitive and low concentration is minutes instead of hours. So we're using this not only for viruses now, we're moving on to measure other things, anything that can be tagged or even intrinsic fluorescence. And with that, I would like to thank my group, my collaborators, our funding. I would just like to mention that we are also recruiting PhD and postdocs. Come to Israel and enjoy fun trips and a lot of science as well. The buffet is waiting for more postdocs. Without you, all of this will just stay in theory in my mind, like this illustration that started the way. So thank you.
Thanks a lot, Eitan. So, questions? Yes, thank you. So it seems like that when you use a dye, maybe you could use actually 2 types of dye. One dye is displaced. Another dye, actually, there's dyes that are intercalating onto the membranes. Then it can be enriched. Then you can have 2 channels just from the dye itself, in addition to your antibody. So you can have more channels to be selective.
You're absolutely right, and we tried that. But one problem that might arise is that we're living in a regime between 400 and 700 nanometer. We can go beyond 700, but it doesn't make much sense with pigments. We can go below, but then we need quartz and not glass, right? So we can squeeze more. I think that multiplexing and adding more signals makes more sense, not necessarily only just by wavelengths, but by different emission ranges, but also maybe adding lifetime capabilities and these sort of things. And we're thinking about them.
Yeah, thank you.
Of course.
Yeah.
A really cool idea. I'm not a doctor, but doesn't mucus and saliva have all kinds of different other stuff in it? So would that obscure your signal, especially in the inverse channel?
Absolutely. So these are usually much larger than my excitation volume. I will see them as huge blobs of no signal, not just dips. So we actually have some preliminary results just from the recent month. It took us some time to get the approvals, to look at mucus of people. And yeah, it's dirty, but it's transparent. So talking about the dirtiness, we have lots of those signals. We have just abrupt reductions all the way to 0 and then come back. Since we did the scaling of what's the size of the dips that we expect for, then basically we know what to expect for. However, we also rely on the dilutionness, how dilute our sample is, right? So if we have too much of those, what we would get is a baseline that is not so stable. And we rely on the baseline to the left and to the right of the signal for the proper quantification of where the dip starts and ends. So we're doing it on the algorithm in which we analyze it, and we still are having a lot of nothingness before and after the signal. So, yeah.
Okay, so thanks a lot, Eitan, again, once more.
Thank you.